There are no questions. Let's begin. Boy, there's only 4 people signed in. That's not very many. All right, can you guys see the screen?
Yes.
Okay, thank you. So if you look on the schedule, we're supposed to be starting chapter 8 today, and we will do that. And then we have a lab later today, lab 8. Note that there is a quiz this Wednesday, and it will cover lab 7 and chapter 5 and 6. And we're going to finish chapter 6 today. We're a little bit behind schedule. Any questions about that? If not, let me move on. And let's get into chapter 6. I discussed this slide here that microbiologists like to plot the growth of microorganisms on a semi-log plot with the x-axis having the time or the generations, and then there's a log of the number of cells, and then when we do that we get a linear curve shown by the dashed line. We can also plot the phases of growth, and this is almost seen all the times in any population you study when you inoculate it or move the population into a new environment. But we're talking about bacterial growth curves, and to my knowledge, you always see this curve with a bacterial growth curve. When you're moving a population from one environment into another, such as taking from a plate where it was grown on solid agar and then you move it into a liquid culture. Initially, when you inoculate that culture, the cells don't grow immediately. There's this lag phase. And they're not growing because they're getting the enzymes they need to grow in the new environment. And once they get those enzymes up and ready, they then start growing. And after the lag phase, we then enter what's called the log or the exponential growth phase, where the cells grow exponentially. And initially, the reason why they're growing exponentially is because there's few cells, there's lots of space, there's lots of resources, and initially there's no waste products. And so the cells just grow exponentially. And then that starts to slow when there's lots of cells, there's not as much space, there's not as much resources, and there's waste products building up. So the growth slows. It eventually slows to the point where we reach the stationary phase. And in the stationary phase, the population number of cells stays about the same. The reason why the population stays the same is that the lucky cells are growing in this stationary phase because these lucky cells, they've got some space around them, they've got some resources around them, and they don't have too many waste products around them. So they're growing. The unlucky cells are dying because they've got lots of waste products around them, they don't got much resources around them, and they don't have much space around them. So in the stationary phase, growth equals death. Any questions about that? And then after the stationary phase, we reach the death or the decline phase. And here, initially, the number of cells dying is greater than the number of cells growing. And so the population starts declining. Now, late in the death or decline phase, you have no cells growing, you only have cells dying. All right, any question about these 4 phases of growth? The lag phase, the log or exponential growth phase, the stationary phase, and then the decline phase. All right, if there's no questions about that, we come to the next couple of slides, and all of this is in your in-class activity, meaning your worksheet for this lecture. So I'm not going to talk about it, but it does discuss the different ways that we can count cells. So how we can measure the growth of microbial populations. There are direct methods, such as the plate count method, also called the viable plate count method. And it's called that because this is the only method that looks at counting viable cells. All the other methods count total cells, including those that are viable and those that are non-viable. There's the filtration method, the most probable number method, the direct microscopic count, meaning counting cells under the microscope, turbidity, the metabolic activity of the cells or population, and then the dry weight. The direct methods being the plate count, the filtration, which what you're doing is you're filtering the cells, and then you're putting the filter on a plate and growing them up. The most probable number and the direct microscopic count are all direct methods. And then the turbidity, the metabolic activity, and the dry weight are indirect methods of counting microbial populations. Just for your information, I've done all of these methods for counting microbial populations except for the most probable number. I've never done this one. All right, any questions about the measurements of microbial growth? Okay, if there's no questions, that brings us to the end.
Is there a reason why you haven't done the most probable number? Is it not as accurate?
It's an arcane mathematical way of getting your estimate. And, well, I'm not real good as a mathematician. I mean, I can do algebra too, but I think, to tell you the truth, the most reason is that most people don't use this method. Okay? I don't even have a slide discussing it. Most people don't use it, and I've just never used it, okay? Because I've had other ways to do it. The quickest way is turbidity. Most people do viable plate counts because you're counting viable cells, or a direct microscopic count because this one's pretty quick if you're looking at cells under the microscope, not as quick as turbidity. One reason for doing the microscopic count is it's a direct method. Turbidity, you're getting an estimate of the number of cells. But no, there's no real good reason. I just never have done the most probable number. And I think most people just haven't used that way. All right. If there's no further questions, let's end this lesson. All the other slides are slides you will look at for the worksheet, so I don't want to talk about them. And then let's move on to lesson 8, which we're supposed to start today. And this lesson covers genetics, microbial genetics. So as always, I have a slide introducing the major goals and the ref lesson outline. Please understand the terms. We'll actually talk about all of them. be able to describe what DNA replication is, what DNA transcription is, which makes RNA, and then the messenger RNA is translated into protein. So understand what protein translation is. And then lastly, understand the different types of mutation. You'll do this in your worksheet. Here's a little bit of a chapter summary. So we're going to start with talking about the structure and function of the genetic material, talking about the flow of genetic information, and then move on to DNA replication, RNA transcription, and lastly, protein translation. And we will get someplace. We'll cover most of those. We might not cover protein translation today, but we'll get at least started, I think, with transcription. Then we'll move and look on regulation of bacterial gene expression, meaning looking at the regulation of genes, and how that's done is with the operons. We will look at mutations, and you'll do that in your worksheet. And we will look at the genetic transfer and recombination. And then lastly, we'll talk about genes and evolution. Any questions about what we're doing? If not, the first term you should know is genetics, the study of what genes are and how they carry information, and then how that information is expressed. We'll look at that, and how genes are replicated. So that's all genetics. A gene is a segment of DNA that encodes a functional product. It usually encodes a protein, but there are some genes that encode RNA, like the tRNA genes and the rRNA genes. And here we're looking at a gene, a sequence of DNA, nucleotides in the DNA, and there would be a second gene. And it is important to realize that the genes don't have to be right next to each other, that there can be regions that are non-coding DNA. And then the genes are on the chromosome. That's just a strand of DNA. And when we call it a chromosome, that strand of DNA is highly coiled, no longer in solution. And there's the chromosome showing you all the genes on the right, which you don't need to know, and then just the positions of the genes. When we talk about genome, we mean all the genetic material in a cell. When we're talking about microorganisms, that's also the genome, is all the genes in the organism. Any questions about that? The study of genomes is called genomics. A genotype is the genes of an organism. The phenotype is the expression of the genes. So when we're looking at the phenotype of this fly, we look at what the genotype is, the genes of that organism. For example, in humans, the phenotype is blue eyes and brown eyes, the color of the eyes. And then blue eyes, the genotype is two genes, lowercase b and lowercase b, encoding for blue eyes. The lowercase just means it's recessive. The brown eyes phenotype has two genotypes. It can be uppercase BB, meaning capital B, and it can also be capital B, lowercase b, because capital B is dominant, so it is expressed, and we see that phenotype. We do not see the lowercase b's phenotype when the genotype is capital B, lowercase b. Any question about any of that? For most animals, this isn't necessarily true of microorganisms, but for most eukaryotes and most animals, you always have two genes for the phenotype. And I should say that's talking about simple genetics, not complex genetics. But if you have a diploid organism, you always have 2 genes for the phenotype. If you have a, what the heck is that called? Haploid organism, meaning it only has 1 set of a gene for the phenotype, and some microorganisms are that way. You might only have 1 gene in the genotype to determine the phenotype. There actually are some human cells which are have only 1 gene to determine the phenotype. Anyone know what those cells are? Come on, those of you at A&P should know. What cells are haploid in the human body?
Muscle?
Say again?
Muscle tissue?
No, those are diploid, but good guess. Thank you for guessing. It's the germline cells, the egg and the sperm. Those are the only haploid cells in the human body. At least I should say the only ones that I know of. When we look at an organism, like a microorganism has a single chromosome, and here you can see it's, when you string it out, it's very long and stringy. This is looking at with an electron microscope, because we could never see this under a light microscope. And that's just 1 strand of DNA making 1 chromosome in E. coli, and on it are all the genes for the E. coli. DNA is a polymer of nucleotides, and you should know them as either A, T, C, or G, or adenine, thymine, cytosine, and guanine. Either way. I don't care how you memorize it. DNA is a double helix molecule, meaning it has 2 strands of DNA, one this way, one that way. The strand of DNA is held together by the sugar phosphate backbone. By that we mean that the strand of DNA is held together by sugar, phosphate, etc. And the nucleotides are important on the DNA, but the nucleotides don't hold the strand of DNA together. What the nucleotides do is they hold the 2 strands together by hydrogen bonds. A hydrogen bonds to T, 2 hydrogen bonds, and C hydrogen bonds to G by 3 hydrogen bonds. You should also know the strands are anti-parallel, meaning one is going this way, the other is going, let me see if I stand up, maybe you can see it. One is going this way, one is going that way. Okay? Any questions about that? We can talk about the flow of information within a cell. The DNA contains the information, so it encodes it in the nucleotides. And that information flows to RNA by transcription. And then the RNA flows to proteins, if the RNA is messenger RNA, by translation. So the information flows from DNA to RNA to protein within a cell. And that's so important in biology that we call it the central dogma, a law in biology, that information flows from DNA to RNA to protein within a cell. Now, I stated that that's the way it happens within a cell, but the information can flow between generations of cells, meaning the parent cell can replicate its DNA, and then one chromosome moves to one side of the cell, the other moves to the other side of the cell, and then the cell splits in two, and we have two daughter cells, each of them getting the parent's DNA. And this is the flow of information between generations of cells, between the parent cell and the daughter cells. If we're talking about humans, the flow of information flows from two parents to one offspring. Any questions about that? The information can also flow between cells of the same generation. And I changed the figure in the book to make it clearer. It's called recombination. But the DNA comes out of this cell, the donor cell. And this cell usually dies. And then the DNA is naked DNA in the environment. And the DNA is taken up by a recombinant cell. And this cell gains DNA from another cell. So if this cell had a gene for antibiotic resistance, and the cell were to die and the DNA were to be in the environment, this cell could take up the DNA and it could take up the gene for antibiotic resistance. Any question about that? The important part is to realize that the flow of genetic information can happen three different ways: within a cell, between generations of cells, or you could say between generations, you're talking about an organism, and then between cells of the same generation, where one cell donates the DNA and the other cell accepts the DNA and becomes the recombinant cell. When we're talking about the flow of information within a cell, you should realize that the information in DNA is encoded in the nucleotides. So that's the language. And there are 4 letters in the alphabet, meaning there are 4 different nucleotides that encode all of the information in the cell. And then that information is passed to RNA, and that information is still encoded in nucleotides. The information is a little bit different because it's RNA nucleotides instead of DNA nucleotides, but it's the same information. And there's also 4 letters in the RNA nucleotide language. Now, when the information is translated from the RNA to the protein in translation, the information is now encoded in the amino acids, and there's 20 different letters in this alphabet of amino acids. And this is true for humans and most organisms. Whoops, didn't mean to move that. And as for comparison, the English language has 26 letters in the language to code the information. Looking at this a little bit differently, when the information is encoded in DNA, the information is "that is a dog." When the information flows to transcription, it's the same information, it's just a different dialect. "That is a dog." Hopefully you could tell the difference in the dialect. But when the information is changed into protein, it's a totally different language, but it contains the same information. Quello è un cane. Any questions about that? All right. If there's no questions about how information flows within a cell, let's talk about how information flows between generations of a cell. The DNA has to get replicated, and then one chromosome moves to one side of the cell, the other moves to the other side of the cell, and then the cell divides into two, and each daughter cell gets the same information as the parent cell. How that happens is the DNA is replicated. One strand, well, the two strands separate, and then one strand is used as a template for making the new strand of DNA. This is called semi-conservative, meaning DNA replication is semi-conservative. When we have the parent strand of DNA, which is black, the daughter cells, each of them will have one dark strand of DNA that came from the parent, and then one strand which is the newly replicated DNA that was made from the parent strand. Any question about that? And the other daughter cell is the same. How the DNA gets replicated is there's a DNA polymerase, and it simply looks at the template strand, meaning the parent strand of DNA, and then sees what this nucleotide is, and it's an A, so the DNA polymerase brings in the correct nucleotide, which pairs with A, and that would be T, because T pairs with A. And then once T is attached to this new strand of DNA, the DNA polymerase then moves down to the next nucleotide, sees that it's C, and then puts in the base that pairs with C, and that would be G, and then that will be attached to the growing new strand of DNA. Any question about any of that? If there's no questions, let's watch a little video on DNA replication. The replication moves in the 5' to 3' direction, so it has to go this way. So one strand can be made replicated sequentially. The other strand has to be done in batches.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letter.
Let me get this louder.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, any questions about the video? There are 3 enzymes you need to know. The helicase splits the DNA and opens it up, separating the 2 strands of DNA. The DNA polymerase adds on the new nucleotides to the growing strand, the new strand of DNA, and the other strand is used as a template for the DNA polymerase. And then the 3rd enzyme you should know is DNA ligase. Whenever you have a strand of DNA, it will not be connected to the other piece of DNA. And the ligase seals that DNA, makes it one single molecule. So what ligase is doing is making a covalent bond in the DNA between this piece of DNA and that piece of DNA. And on the Okazaki fragment, there's going to be lots of broken pieces of DNA, I don't know how to word it other than broken pieces of DNA, that will need to be connected with the ligase. With the leading strand, there will only be a few places where the ligase has to start. Where the helicase first opened up the DNA, and then the DNA polymerase started going, so that would be like right here. You do need to seal where it started to the old piece of DNA over here, and so the DNA ligase will need to work occasionally on the leading strand, but on the lagging strand it's going to be working on each end of each Okazaki fragment. Any question about that? Alright, hopefully you understand the leading strand. The DNA polymerase can read it directly and just move sequentially, but on the lagging strand you have to do it in batches because the DNA polymerase moves 5' to 3', meaning it moves this way and the helicase is moving that way. So that's why we have the Okazaki fragments. If you wonder about why it's called Okazaki fragments, it's Dr. Okazaki who discovered it. Alright, let's look at DNA replication in a little bit more detail. It's just going over a little bit what the movie or video discussed. The new DNA is made in the 5' to 3' direction. It's initiated by an RNA primer. The leading strand is synthesized continuously. The lagging strand is synthesized discontinuously, resulting in several or many Okazaki fragments. The RNA primers are removed from the DNA, both from the Okazaki fragments as well as from the leading strand. And then that's filled in with the DNA polymerase. And then the places where the DNA are broken, meaning they're not connected, the DNA ligase goes in and makes a link, a covalent chemical bond. Any question about that? If not, let's move on to transcription. This is the synthesis of RNA from DNA. When the DNA is transcribed, only the DNA that encodes a gene is transcribed. When we're talking about DNA replication, all of the DNA will be replicated. But in transcription, only the DNA that encodes a gene will be transcribed. And there are regions of DNA that do not encode a gene. In eukaryotes, actually that's the majority of the DNA. And we're not going to get into what that DNA does, because obviously it's holding the genes together. But it probably plays a role, meaning the non-coding DNA, it probably plays a role in the folding up of the DNA to go from a single strand of DNA in solution to one that's highly folded up that we see as a chromosome. And that's just for your own information. The important part is that only the genes are transcribed, not the non-coding DNA. And when DNA is transcribed, we make RNA, and there are 3 types: messenger RNA, tRNA, and rRNA. And here's a question for you. How does the RNA polymerase know which RNA to make? How does the RNA polymerase, when it's reading the DNA, know which RNA to make? When should it make messenger RNA? When should it make tRNA, etc.? Anybody have a guess? I think that's a question on your worksheet, so you should think about that. All right? We'll come back to it, but I want somebody to guess. Transcription begins when the RNA polymerase binds to the promoter. The promoter is a region of DNA upstream of the gene. And what the promoter does is it codes for the binding of the RNA polymerase. The RNA polymerase then proceeds in the 5' to 3' direction on the DNA, and it will transcribe the DNA into RNA. So RNA is made in the 5' to 3' direction. And then that RNA polymerase keeps transcribing the DNA until the transcription until the RNA polymerase reaches the terminator. The terminator is a region of a gene that encodes for the end of the gene, meaning the terminator tells the RNA polymerase stop transcribing. Okay? So every gene, at least every gene that I'm aware of, has a terminator at the end of the gene, and the terminator tells the RNA polymerase, stop transcription. Any question about any of that?
I got a question about the 5-3. I still don't know, even though I took that class last quarter.
Uh-huh.
I still very confused in the 5-3 direction. I mean, it's
Uh-huh.
like 5-3 inches or 5-3.
What is the 5-3 direction? I understand the question. Let me see if I can find a picture to better describe this. I'm afraid I don't have a picture of DNA where the DNA is actually labeled. All right. So this is the 5' end, this is the 3' end. The chemist simply labeled the carbons on the sugar, and this carbon here is the 5th carbon. And don't ask me how they do the count, because I don't know where it is. And this carbon here, right there, is the 3' carbon. Okay? And
Okay.
I don't, oh, here's the 4th carbon, it's right there. I was saying, where's the 4th? So here's carbon 3, so this must be carbon 1, carbon 2, carbon 3. And so this is the 3' end, and there's carbon 4 right there, and here, right there, is carbon 5. Okay? It's just a way for biologists or chemists to know which carbon they're talking about in the deoxyribose, the sugar right here. All right?
Okay, thank you.
You got it? Good. You won't be tested on that, by the way. But it's just what it is. You should know that the DNA runs opposite of each other. Let me see if I can see where that is. Ah, here we go. Where one is running this way and the other one is running the opposite direction. Okay, we were on this slide, and I think we finished it, and that is the RNA polymerase binds to the DNA at the promoter and then starts transcribing the DNA to make RNA. And that continues until the RNA polymerase reaches the terminator. The terminator codes for the RNA polymerase to fall off the DNA. This is actually looking at the RNA transcription happening in an actual cell of a this isn't an electron microscopic image. It's actually, I think, an antibody fluorescent microscopic image. All right, let's go through transcription in a little more detail. So here's the gene. Let me blow that up. Here is the gene. And the promoter is right here on the DNA. And that's where the RNA polymerase will bind to the DNA. And then it just moves down the DNA, copying the DNA to make RNA. And where the polymerase binds to the DNA, we will have a single strand of DNA. So the DNA will actually be single-stranded here. And there is a helicase that opens up the DNA, making it single-stranded. And then the RNA polymerase binds, and then it starts making RNA from the DNA. And only one strand of the DNA is transcribed. Everyone got that? And it's the strand of DNA that encodes the information in the gene. This region here will be antisense for the genetic information of the gene, and it is not transcribed. Any question about that? And then the RNA polymerase just moves down the DNA, transcribing the DNA and making the RNA. And the RNA is made because it'll be the corresponding nucleotide to the DNA. And what's the word I'm looking for here? When you read A, the RNA polymerase will put in U. What's the word I'm looking for there? What is that? It's the...
Uracil?
Yeah, but it's the RNA base that binds to A. I'm trying to remember what the heck that's called. Not the antisense, the corollary. Okay, so U will be put in for against A, G will be put in against C, A will be put in against T. The corollary nucleotide. That's what I was looking for. And then the terminator just keeps doing, not the terminator, the RNA polymerase just keeps doing that. It moves down the gene, transcribing it into RNA, and it continues doing that until the RNA polymerase reaches the terminator. At the terminator, that codes for the RNA polymerase to fall off, and when the RNA polymerase falls off, that releases the RNA, and then it can be used by the cell to make whatever the cell wants to make. Like if this is a gene encoding a protein, this will be a messenger RNA, and it'll be used to translate the gene into protein. Okay? And that comes back to the question, how does the RNA polymerase know which RNA to make? How does it know to make messenger RNA? How does it know to make tRNA? How does it know to make rRNA?
The codons?
Yeah, what the codons are, meaning if this gene codes for a protein, then it will make messenger RNA. But if this gene codes for tRNA, it will make tRNA. And if the gene codes for rRNA, it will make rRNA. It's that simple. It's whatever the gene codes for is the type of RNA that will be made. Any questions about any of that? All right, if there's no questions, let's watch a little video on RNA transcription. Let me see if I can open it. Oh, that took a long time. Oh dear. Let me try well, I'm at YouTube. Let's go ahead and look for one. That isn't too long. I think this is the one that I want. Or maybe it's that one. No, it's this one.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex.
Okay, we don't need to know about that. All right, any questions? Let me see if this link works. We won't watch it because we've already watched a video, but I just want to see because that link I've got to replace. Yeah, both of those links don't work. Let me make a note of that and I'll correct that. Oh, that's not good. This is odd. Why isn't that showing? All right. Let's move on. I think we've already talked about that, so I'm not going to talk about it further. So, if messenger RNA is made, the messenger RNA in a eukaryote cell will leave the nucleus and go into the cytoplasm. In a prokaryotic cell, the messenger RNA is already in the cytoplasm, and in the cytoplasm, the messenger RNA can be translated into protein. The synthesis of proteins by the ribosomes happens using 3 of the rRNAs, meaning the messenger RNA encodes the information for making the protein, but the rRNA is needed and makes up part of the ribosomes. Remember, ribosomes are rRNA and ribosomal protein. And that's what is the protein factory for making the protein, meaning the ribosomes. And then the third RNA is also important. The third RNA is tRNA, and it brings in the nucleotides, not the nucleotides, the amino acids for the growing protein. And tRNA is actually the one that reads the information on the messenger RNA. And we'll talk more about that in a minute. The messenger RNA is translated into protein, using codons. A codon is simply a group of 3 amino acids. So when we're translating our message into proteins to determine an amino acid, you read 3 of the nucleotides. And that we call a codon. So in making of the amino acids, you don't read one nucleotide one after the other. You actually read them in groups of codons, or 3 nucleotides. Any question about any of that? Okay, translation of the messenger RNA begins at the start codon. There's one codon that codes for the start, and on the messenger RNA there will be this codon, and it just tells the ribosome, start protein translation here. And that codon, you should probably know, is the codon AUG. So on the messenger RNA, the nucleotides, or the codons, whichever you want to talk about, before the AUG code for nothing in the protein. Everyone got that? Protein translation doesn't start until the ribosome comes to the AUG on the messenger RNA. Usually the AUG is near the start of the messenger RNA. It's not at the start, but it's near the start. But it can be down further, in which case there will be a portion of the messenger RNA that does not code for protein. And then the ribosome moves down the messenger RNA to translate the messenger RNA into protein, meaning amino acids. And it continues doing that until the ribosome reaches one of the 3 stop codons. So it'll move down the messenger RNA until it reaches a stop codon, and there's 3 of them. You do not need to know the nucleotides that code for start, but if the ribosome hits UAA or UAG or UGA, the ribosome will stop protein translation. Any question about any of that? All right. So each messenger RNA codon codes for 1 amino acid. Organisms in general have 20 different amino acids. And this is true of humans, and it's true of most bacteria. Okay? But remember, we have 4 different nucleotides, and those 4 different nucleotides can go in 3 different positions. That means we have slightly over 60 different codons. And there's only 20 different amino acids in most organisms. And that means that each codon can code for more than, no, that's not quite the way to word it. Each amino acid can have more than 1 codon coding for it. So if I'm talking about a codon, there can be several codons that code for the same amino acid. There are a few that only code for 1 amino acid, like AUG, the start codon, does code for start, but it also codes for the amino acid methionine. So AUG has a dual function. It codes for start, starting protein translation, and it codes for the amino acid methionine. And by the way, that means that whenever you start a protein, you always start with the amino acid methionine. So every protein that's made has at least one methionine in it. And if the protein doesn't want to start with methionine, it'll have to remove that methionine. Because in protein translation, it'll always have methionine first, because that's the start codon. Can anyone see it? There is one, here it is here. The other amino acid, you don't need to know this, but there is one other codon that only codes for one amino acid, and that's UGG, codes for tryptophan. All the other amino acids have more than one protein coding for them. Some, like phenylalanine, only have 2 codons coding for that amino acid, but others, like leucine, have 6 different amino acids, not 6 different amino acids, 6 different codons coding for the same amino acid. Did everybody get that? I sort of made a blunder when I was talking. So this means that there is more than one codon for each amino acid. And this is known as the degeneracy of the code. And this is not meaning like a degenerate person, a despicable, low person. It means degenerate in the sense that there's more than one message that codes for the same thing. And there's only one codon that codes for start, and it also codes for the amino acid methionine. So remember that. Students have trouble with that. AUG does code for start, but it also codes for the amino acid methionine. But there are 3 codons, which you don't have to memorize, that code for stopping the protein translation. All right, any questions about any of that? If not, let's go into protein translation a little bit further. And I think we'll start with the video.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon.
Okay, you don't need to know the names of the different sites to the ribosome, but you should know that there are 2 sites in the ribosome.
The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, any questions about the video?
No.
Okay.
Attack acid. The large cell ribosome now binds to create the peptide or P site.
I'm looking for a place. Here it is.
The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then...
Okay, so what removes the amino acid here and then connects it to the amino acid here, that amino acid to there, that is an rRNA enzyme. It's the ribosome that does that, and it's an rRNA enzyme. This is the only enzyme we'll talk about that is an RNA enzyme. Any questions about any of that?
Is that the same as a messenger RNA?
No, this is the messenger RNA down here.
Okay.
I'm talking about the only enzyme that we'll talk about that is an RNA enzyme. What it does is it removes this amino acid from the tRNA and then links that amino acid to this amino acid. And that is the only example of a RNA enzyme that we're going to discuss in this class. Okay, so I just wanted to show you right there. All right, let's close this video. Boy, I've got a lot of these here. And then go on the slides discussing the from messenger RNA to protein. Hopefully I didn't change that there. So when DNA flows to messenger RNA, that's transcription, and then when messenger RNA flows to protein, I'm talking about the information of the genetic information flowing. When it flows from messenger RNA to protein, that we call translation. Translation includes the ribosomes, and there are 2 subunits, the large subunit and the small subunit. The messenger RNA, which codes the information from the DNA, and then the tRNA, which brings in the correct amino acid. And the tRNA is also important because it's the tRNA that's reading the messenger RNA. So for example, how we know that AUG codes for methionine is the tRNA actually binds to AUG with the anticodon UAC. And so the tRNA is translating this message into the amino acid. Any question about any of that? But it all happens at the ribosome, so we call that the factory, and the messenger RNA brings the information from the DNA out to the factory. And I guess the tRNA in this analogy would be the workers in the factory, who, if you didn't have them, no work would get done in the factory. Any questions about that? So the first TNA comes in the first site of the ribosome, and that's all you need to know of it. And protein translation happens at the codon AUG because that's the start codon. The first tRNA will have the anticodon for AUG, meaning that will be UAC, which you don't need to know, but it's the corollary of AUG. And it will bring in the amino acid methionine. So all proteins start with the amino acid methionine. And if we don't want that protein to start that amino acid to start the protein, it can be cut off the protein. And then the protein will start with a later amino acid. But we're not going to talk about that. And then the next tRNA comes in the second site in the ribosome, and it will have the corollary basis to the next codon. So UUA is the next codon, so AAU will be the anticodon on that tRNA, and it'll bring in the correct amino acid. Okay, then an enzyme removes the first amino acid from the tRNA and then links it to the second amino acid to start the growing polypeptide chain. And at the moment we only have two, so we'll just call it a peptide chain. All right, any questions about any of that? Once that happens, the tRNA is released and it falls out of the ribosome. And then the ribosome, when this is empty, moves down one position. So what used to be in the second position of the ribosome moves now to the first, and then this is open and the correct tRNA comes in, bringing the correct amino acid. The enzyme that we talked about earlier removes the amino acid from this tRNA and then attaches it to that amino acid, and that means that this tRNA can then fall out. So it falls out, and then the ribosome moves down one codon. And this process continues that way until the ribosome reaches the stop codon. Once it reaches the stop codon, what the stop codon codes for is the stop protein translation. And the video talked about it in a little more detail. We're not going to go into that much detail. But no further amino acids are added. The polypeptide is released, and now that's the finished protein. The tRNA falls off of the ribosome, and the ribosome falls apart, and that releases the messenger RNA. And then this messenger RNA can be used by another ribosome, or maybe the same ribosome, to make another protein. Any question about any of that? All right, let's talk about a complication. In prokaryotic cells, we have two complications. In eukaryotes, one of the complication doesn't happen, and we'll discuss why in a minute. But in prokaryotic cells, we can have the DNA being translated as messenger RNA, and then the protein can be made in the cell. And as you can see from this picture, there are many ribosomes, 1, 2, 3, 4, 5, 6, 7, 8 ribosomes on this messenger RNA, and that's an actual electron microscopic image. And this is the artist's rendition, reading that messenger RNA. And this can happen in both prokaryotes and eukaryotes, where the messenger RNA will have more than one ribosome on it, and so there will be several growing polypeptides on the messenger RNA. Any question about that? In prokaryotes, it can even be more complicated, and this is actually what this is showing. This is the strand of DNA, and the messenger RNA is being made, and so there's longer strands of messenger RNA on this end compared to that end. And in prokaryotic cells, the DNA is in the same place as the RNA, the cytoplasm, and then the RNA can be translated into protein. And so here in this picture, we got the DNA and the RNA polymerase making the messenger RNA, which is the blue line. And then we have the ribosomes attaching to the messenger RNA, and this ribosome has this messenger RNA has 5 ribosomes attached to that messenger RNA, and the messenger RNA is still being made off the DNA, meaning in prokaryotes you can have protein translation starting before RNA transcription has finished, because it's all happening in the cytoplasm. Any question there? Now, you don't have that in eukaryotes, because transcription happens in the nucleus and then translation happens in the cytoplasm. But you can have more than one ribosome on a messenger RNA in eukaryote cells. And so you just need to know these complications. Okay? Any question about any of that? If there's no questions, let's end this lecture, and I will see you in the lab at 6:30. Okay? Oh, fudge.